Hello, everyone. Welcome to today's program. I am happy to have you all here today and looking forward to getting started. So my name is Jamie Seward. I'm an Associate Director of Affinity Engagement Programs with the Office of Alumni Relations here at Johns Hopkins University. Before we proceed, I'd like to acknowledge our co-sponsors, the Department of Neurology and Brain Sciences and the Healthcare Affinity. Now, before I turn the program over to our moderator, I encourage you to ask questions by typing them in the Zoom Q&A at the bottom of your screen. Now, it is my pleasure to turn the program over to our moderator of Neurology and Brain Sciences. Rachel, it's a pleasure to have you here today.
Thank you, Jamie, and thank you everyone for joining us today. I'm just going to go ahead and introduce our first speaker, Dr. Sarah Berth. Dr. Berth is an assistant professor in neurology that specializes in neuromuscular disorders. Dr. Berth's research interests include understanding cellular mechanisms for degeneration in neuromuscular disease. She is particularly interested in understanding how transport of organelles and autophagy, clearance of damaged proteins, is disrupted in ALS. Oh, I think you're on mute.
All right, so let me just share my presentation. Okay, can you see it? I'm gonna assume yes. Okay, great. Thank you, Rachel, for the introduction, and thank you, everyone, for your attendance today. I'm Sarah Berth, and I'm starting my second year as assistant professor at Johns Hopkins in neurology. I specialize in neuromuscular disease and see patients with neuromuscular disease such as ALS. But another major focus for me is to understand the mechanisms underlying neuromuscular disease with a goal to find new treatment targets. To do this, I spent a lot of time doing basic science lab experiments also, and I'm excited to tell you about one of my projects regarding ALS today. All right, so Amyotrophic Lateral Sclerosis, or ALS, is a devastating neurodegenerative disease. It primarily affects motor neurons, which are the neurons responsible for controlling muscles in the body, and it causes progressive degeneration and death of these motor neurons. Now, ALS typically starts with weakness in one area of the body, for example, a part of an arm or leg, and then the weakness gradually worsens and involves more and more muscles, and then spreads to other limbs, causing progressive paralysis. So what you can see in this diagram is that symptoms can include weakness, atrophy or loss of muscle mass, stiffness, and cramps in muscles. But note that motor neurons also control the muscles for speech and swallowing, and most devastatingly, they also control the muscles for breathing. But this means that people with ALS also develop difficulty with speech and swallowing food and liquids, and they eventually develop progressive difficulty breathing. Now, ALS is a fatal disease with average mortality of only 2 to 5 years. Current treatments are really only minimally beneficial, and there is no cure. Finding effective treatments for ALS is less of paramount importance. Now, the majority of ALS cases are sporadic. However, close to 10% of ALS is familial due to a number of different genetic mutations that are passed down in families. Studying mechanisms by which gene mutations can cause ALS could provide insight into mechanisms for development of sporadic ALS. Now, the most common genetic mutation that causes ALS is a hexanucleotide repeat in a non-coding region of the C9orf72 gene. Large expansions of this nucleotide repeat cause ALS. So you can see that these C9orf72 mutations in red make up a small percentage of sporadic cases, as well as about 40% of familial cases, making this an important mutation to study. We are focused on understanding the cellular mechanisms for how this genetic mutation causes ALS. Now, out of all the cells, and even out of all the neurons in the body, we know that motor neurons are selectively targeted in ALS. So what is it about these motor neurons that makes them uniquely vulnerable? Now, here's a diagram of a neuron. Here's a cell body or soma, where the nucleus resides and where almost all proteins are synthesized. And here are the dendrites, which are responsible for receiving information from other neurons. Now here's the axon, which is important for transmitting information. The axon terminals release chemical neurotransmitters to communicate with other cells. Now, motor neurons are unique because they connect with and control muscle through large bundles of axons, which make up their nerves. To achieve this, they have a specialized structure. Now, here is a motor neuron drawn to scale. So over here is a cell body in the dendrites, and over here is an axon terminal. And this entire length here is comprised of the axon. And you can see here that a major unique feature of motor neurons that may convey their selective vulnerability is that they're remarkably polarized. So the cell bodies of motor neurons are 100 micrometers, or only 0.1 millimeters in diameter, and they're located in the spinal cord. These cell bodies then send their axons to innervate a muscle, and then their axon terminals will release neurotransmitter to activate the muscle. Now you can imagine that if this motor neuron has to interact with a muscle far away from the spinal cord, for example, say a muscle in the foot, then the axon needs to be remarkably long. And in fact, motor neuron axons are up to 1 meter in length. Now compare that to the 100 micrometer size cell body. Now this polarity poses a huge challenge for neurons. The fascinating thing is that virtually all protein synthesis occurs in the cell body. So almost all proteins necessary for maintenance of the axon and important for synaptic transmission are synthesized in the cell body, and then they need to be transported with precise timing and to the correct location within the axon for the neuron to survive. And this process is a highly regulated process called axonal transport. Now, axons have parallel microtubule tracks, shown here in gray, running along the axons. And I think of these as like highways along which necessary proteins are carried and delivered. This is done by motor proteins kinesin and dynein, which walk along these microtubule tracks using ATP for energy and carry proteins from the cell body to the end of the axon and in the reverse direction. And you can imagine that if this process is disrupted, just like if there's a car accident on a busy highway, then this will lead to deleterious consequences for the neuron, including axonal degeneration and cell death. Now, we know that axonal transport is disrupted in ALS. Even some of the earliest studies looking at postmortem tissue showed swellings in axons with accumulation of proteins, showing that these proteins were not getting transported as they should have been. Defective trafficking is commonly seen as the common theme across multiple ALS models. And in fact, underscoring the importance of proper function of axonal transport to prevent ALS is that genetic mutations in motor proteins themselves can cause ALS. Now, axonal transport is not the only disrupted pathway in motor neurons in ALS. And in fact, another cellular mechanism that's dysregulated is clearance of proteins, or autophagy. Now, when proteins are destined to be degraded or recycled, they're tagged and then targeted to membranes that actually engulf these proteins and form structures called autophagosomes. Then the autophagosomes fuse with lysosomes and then mature into autolysosomes, which ultimately degrade the proteins inside them. In ALS, motor neurons have an accumulation of proteins, indicating that proteins aren't getting degraded and cleared how they should be. And in fact, again, genetic mutations in autophagy proteins can also cause familial ALS. But how are these two separate cellular processes interrelated? And in fact, recent research has shown that autophagosomes, which remember are these organelles in which a membrane is enclosed around the protein to be degraded, but these autophagosomes are in fact generated in the distal axon, and then they actually mature as they are retrogradely transported to the cell body. Once they are in the cell body, they're the final mature autolysosome and can degrade proteins inside them. So it's now known that autophagy and axonal transport may be interrelated, and disruptions in one process may affect the other. So in pathogenic conditions down here, so impaired autophagy could lead to the accumulation of autophagosomes or lysosomes in the axons, which could then impair axonal transport of other proteins. But alternatively, impaired axonal transport could lead to protein accumulations in axonal spheroids that then prevent or slow down autophagosome transport, which could ultimately affect autophagy. And this is a pathway I'm very interested in examining and parsing out in ALS. Now, Drosophila are a useful model for studying genetic underpinnings of disease. And this is because genes of interest can relatively easily and quickly be introduced and expressed under a promoter using a GAL4 UAS binary driving system. In fact, we can express genes of interest in specific tissues, in our case, specifically in motor neurons. In flies, a well-established marker of neurodegeneration is degeneration of photoreceptor neurons and their surrounding glia, which leads to a change in externally observable morphology, which can then be scored and quantified. And in fact, when we express the expanded C9 or 72 hexanucleotide repeat in flies, we can see eye degeneration here, and then we can also see decreased survival. Now, a postdoctoral fellow in the Lloyd Lab, Hyun Sung, has looked at axonal transport in Drosophila, expressing the C9 or 72 expanded mutation in motor neurons. When he labeled autophagosomes, so shown here in red, he found a reduced amount of autophagosomes being trafficked in the axons. So here you can see the chimegraphs, which are plotting movements over time. So if the organelles don't move, then you have a vertical line. If they move anterogradely or to the axonal terminal, then their position moves to the right over time. And then retrograde motion will show position moving to the left over time. And so you can see here that compared to control, and these flies expressing the expanded repeat, there are clearly less autophagosomes being trafficked. And these changes are specific to the autophagosomal pathway, because if you look, for example, at moving mitochondria in green down here, then you don't see a difference. What about later in the autophagosomal pathway? Here, Hyun looked at lysosomes moving in the axons and found in these bottom two panels that there were more lysosomes in the axons, and that there were also more lysosomes that weren't moving, indicating a perturbation in the process of autophagosomal transport in axons. Now, I've begun work on expanding these findings to a human model of C9 or 72 ALS. And I wanted to talk a little bit about induced pluripotent stem cells. Now, pluripotent cells are precursor cells that can propagate indefinitely, as opposed to neurons, which do not divide and propagate. And pluripotent cells give rise to every other type of cells. The most well-known type of pluripotent stem cells are embryonic stem cells. These have been used in science, but have been controversial because they require manipulation of a pre-implantation stage embryo to make them. Now, induced pluripotent stem cells use a newer technology and avoid the use of embryos. Basically, you can obtain a fully differentiated cell from someone, for example, a skin cell or a blood cell, and then activate certain genes to actually reprogram the cells back into stem cells. And in this case, blood samples were obtained from patients with the C9 or 72 ALS mutation and controls. These blood cells were then reprogrammed into induced pluripotent stem cells, or iPS cells, and then had a gene introduced to turn on differentiation into neurons, as you can see in this image below. Now, this is a powerful tool because then we are utilizing cells derived from people who have ALS. Now, what we are working on in these cells is growing them in microfluidic nanofabricated devices that have grooves that are long enough that only axons can grow through to the other side, but small enough that cells can't get through. So we are in effect isolating axons. So you can see that in this diagram. So we can plate the cells on one side, and then their axons grow through these grooves to generate annexonal compartments. So we are visualizing axons at the end of these grooves. This makes the axons nicely aligned. So we know which direction is anterograde or away from the cell body and which direction is retrograde or towards the cell body. Then we are performing real-time imaging of different proteins in the autophagy pathway to really try to work out how axonal transport is affected in C9 or 72 ALS, and then how we can regulate and rescue it. So you can see below, here is a video that I've taken examining lysosomes, which are in later stages of the autophagy pathway in human iPS cells. So lysosomes are labeled with a red fluorescent dye. And here you can see the axons within a microfluidic groove. And moving to the right here is anterograde or towards the axonal terminal, while moving towards the left is towards the cell body. So this is an experiment using real-time imaging, the super resolution microscopy, to evaluate lysosome axonal trafficking. So with these movies, we can quantify velocities, number of lysosomes, number of stopping and starting events to really get details about axonal trafficking in these neurons. So how do we explain the decrease in autophagosomes, but the increase in lysosomes? Now, what we think is happening is that there is decreased autophagosome biogenesis in C9orf72 ALS. Because of this decreased number of lysosomes, less are available for fusing, because of this decreased number of autophagosomes, less are available for fusing with lysosomes to mature into autolysosomes. So then there's this accumulation of lysosomes that aren't getting used. Now, I'm working on evaluating axonal trafficking of different parts of the autophagy pathway in iPS cells to further evaluate how this pathway is affected in C9orf72 ALS. Then my goal will be to modulate the pathway to rescue any deficits to prevent axonal denervation and degeneration that's seen in ALS, with the goal to define new, more effective treatment targets for ALS. All right, and that's it. I wanted to acknowledge several people. First, I want to thank my mentors and collaborators. So I'm extremely lucky to be in such a collaborative atmosphere at Johns Hopkins. I'd like to especially thank Dr. Lloyd, who is my primary mentor and has been really fantastic to work with, as well as Dr. Rothstein, who's been an incredible mentor for me. I also want to thank Dr. Hayes, who will be speaking soon, who was really helpful for me when I first started this project. And then in terms of collaborators, I've worked with many of the members of the Lloyd and Rothstein lab. I want to especially thank Hyun Sung, who I've worked closely with on this project. And then I also want to thank Mohammed Farah, whose lab has designed the microfluidic devices I've been using, and also Shu-Ying Sun, whose lab has expertise with the IPS neurons I'm using and I've been collaborating with regarding these cells. Then finally, I want to thank NIH, specifically National Institute of Neurological Disorders and Stroke, for supplying me with funding for my research. With that, I'd like to thank you for your attention and take any questions.
Thank you so much, Dr. Berth. We do have at least one question. I encourage everyone to put your questions into the Q&A, but we can start with the first one. What is the link between C9 and autophagy lysosomal pathway? Do you hypothesize that C9 plays a role in this pathway?
Yeah, that's a really good question. And so we know that that C9orf72 that it does play a role in autophagy. But we actually think that in these mutations, that it's more of a gain of function as opposed to loss of function. And so we think that what this expanded hexanucleotide repeat is doing is actually impairing nuclear transport, which I haven't talked about, but which Dr. Hayes will be talking about soon. And that this prevents transcription factors that regulate autophagy from being able to turn on the genes to activate autophagy.
Great, thank you so much. We just got another question. Do all of the motor neurons eventually die off, or do remnants remain? Why does the disease generally occur later in life?
Oh, interesting. Okay, so they not all the motor neurons die off. So basically there's an axonal degeneration first, and then over time, more and more of them die off. But we know that some motor neurons aren't affected, which is really interesting. So for example, the motor neurons that control eye movements actually, remain unaffected, and that actually is really helpful for ALS patients because they can communicate. When they start having difficulty with speech, they can use eye tracking, to communicate. But we're not really sure what it is about those neurons controlling eye movements that makes them unaffected. But that's a really interesting area of research. And sorry, what was the second question? I got excited talking about eye neurons.
Why does this disease generally occur later in life?
Yeah, that's a really good question too. So we think in neurodegeneration. So let me take a axonal transport centric approach. So neurons have more axonal transport than what is actually needed, but we know that over time that this fast axonal transport, the rates decrease and less is transferred over time. So say say in some of these mutations, compared to say this is a normal amount of transport and it kind of goes down, and then here is less, but that's also declining over time. And then at some point you reach the set point where you start actually affecting the motor neurons.
Okay, great. So I'm going to now introduce, thank you so much, Dr. Berth, and I'm going to introduce our next presenter, Dr. Lindsey Hayes. Dr. Hayes is an assistant professor of neurology and a neuromuscular specialist. In 2020, Dr. Hayes established her own independent laboratory with a focus on mechanism and regulation of TDP-43 nuclear shuttling and mislocalization, a major pathologic hallmark of sporadic and genetic forms of ALS and FTD, frontotemporal dementia. By understanding the mechanisms governing TDP-43 localization and function, she hopes to identify novel therapeutic targets for ALS and FTD. Turn it over to you, Dr. Hayes.
Great. All right. I think I have my slides up, so hopefully everyone can see that all right. Thanks very much for the opportunity to present today. I'm very grateful to be here and for everyone that's tuned in and for the opportunity to share my work with a broader audience. Like my colleagues in this seminar, I'm a neuromuscular specialist, so I see patients with ALS and other disorders of nerves and muscles. And I run a neurobiology lab that studies the disruption of RNA and protein trafficking in ALS, otherwise known as Lou Gehrig's disease, after, of course, one of the famous early patients with this terrible disorder. So my group is specifically focused on the mislocalization of an RNA binding protein called TDP-43. And this is a major pathologic hallmark of ALS, as I'll discuss today. We're working very hard to understand what causes TDP-43 to leave the nucleus of cells and to develop therapeutic strategies to target TDP-43 mislocalization. So I won't spend a lot of time because Sarah's already done such a nice introduction. ALS is a fatal degenerative disease of motor neurons in both the brain and the spinal cord, as marked here in green, that progressively robs patients of voluntary movements. It's relentlessly progressive. The mean survival is about 2 to 5 years. It varies a bit on depending on the type of onset, as you can see, and our treatments only modestly slow this decline. Although it's considered to be a rare disease, the estimated cumulative lifetime risk of ALS in the United States is about 1 in 400 people, which really emphasizes the urgency of better understanding this disorder and developing more effective treatments for this disease. There's been major progress in the past 30 years identifying mutations in that 10% of patients Sarah mentioned that have inherited ALS or familial disease. And you can see that discovery rate since the first discovery of SOD1 in the mid-90s. And these mutations cluster along many pathways, one of which is RNA binding proteins like TDP-43 itself, which I'll discuss more, protein homeostasis and cytoskeletal proteins, and obviously Sarah already mentioned the most common mutation, C9orf72, which accounts for about 1 in 12 cases of ALS overall. Still, approximately 90% of cases of ALS in patients without a family history, so we call this sporadic disease. The cause of ALS in these patients remains unknown. And this is one of the most common questions that I get in clinic when I'm giving someone this terrible diagnosis. They want to know, why is this happening to me? And unfortunately, for the bulk of ALS patients, we still don't have an explanation. Nevertheless, encouragingly, there are 3 different antisense oligonucleotides in clinical trials for the mutations that I've indicated here. We still are accumulating data on whether or not this will be effective, but the goal is to try to turn off these genes. And obviously, that is an approach that many of us are excited about following. So despite all of the variability in the clinical presentations that I mentioned and the different mutations, pathologically, about 97% of ALS patients show the same finding, which is this localization of this RNA binding protein TDP-43. It's also seen in about half of patients with this genetically related disorder called frontotemporal dementia, which affects slightly different areas of the brain, but shares a lot of clinical and pathologic overlap with ALS. TDP-43 normally resides in the nucleus of the cell. This is this membrane-bound hub where all of our DNA is located and transcribed into RNA. Like many RNA binding proteins, TDP-43 shuttles back and forth between the nucleus and the cytoplasm through a process known as nuclear transport. We know that TDP-43 is critical for RNA processing in the nucleus, and quite a bit has been discovered about how loss of that normal function of TDP-43 is detrimental to neurons, and how accumulation of TDP-43 in cytoplasmic aggregates within cells can also cause cellular toxicity. Based on these data, TDP-43 mislocalization is thought to be integral to the pathogenesis of ALS and also an important therapeutic target. However, the upstream cause of TDP-43 mislocalization in disease remains unknown. So our primary focus over the past year or two has been to determine the mechanism and regulation of TDP-43 nuclear export, even in healthy cells. This is something that's historically not been well understood, and it's really critical to tease apart the cause of TDP-43 mislocalization in disease. So I'll tell you more about this in a moment. And we've recently submitted this, it's in review, and for those interested, a preprint is available in bioarchives. So armed with this better understanding of what regulates TDP-43 nuclear export, we're now moving on to some more complex disease models to try to understand the cause of mislocalization in disease and to try to develop RNA-based therapeutics to target TDP-43 mislocalization. So a few slides on the export studies I mentioned. TDP-43 undergoes nucleus cytoplasmic shuttling between the nucleus and the cytoplasm of the cell. It's very well established in terms of the mechanism by which TDP-43 enters the cell. In terms of nuclear export, though, it's a lot less clear. It was long thought to utilize this export receptor called exportin-1, but a few years ago, that was shown not to be the case, leaving open a number of other potential candidate mechanisms by which TDP may leave the cell. And so we really wanted to develop nuclear transport assays to answer the question of how TDP-43 leaves the nucleus and how that is regulated. And to expedite this type of work, which involves a lot of cell culture and some pretty tedious image analysis, we, for the past several years, have gone entirely to something called high content microscopy. It's this rather boring looking box that you see over here, but it houses a high-speed spinning disk confocal, and that allows us to very rapidly image cell lines and neurons plated in these 96-well plates. And then we can apply automated algorithms to look at the translocation of fluorescent probes in and out of the nucleus of cells, and in doing so, capture thousands of cells per well or treatment condition in a very rapid and unbiased manner. And so this approach of high content microscopy has really accelerated the type of screening and discovery that we can do in the lab. So here's an example of the type of experiments that we can do with this approach. It's been known since the early 90s that when you inhibit RNA synthesis, you can shift TDP-43 and other RNA binding proteins into the cytoplasm. And that's demonstrated here with an mRNA synthesis inhibitor. And we can use our automated quantification to look at this marker for newly synthesized RNA together with TDP-43. And you can see that as we inhibit RNA synthesis and there's less RNA in the nucleus, TDP-43 spills into the cytoplasm in a dose and time-dependent manner. And the mechanism of this was never really clearly delineated. It was generally thought to reflect the idea that RNA binding proteins follow the gradient of RNA within cells. Then what the high content allowed us to do was go on and compare about 25 other RNA binding proteins to TDP-43 in terms of how quickly they respond to inhibition of RNA synthesis in cells. And this result was very informative. So you can see that compared to a panel of about 25 other proteins, TDP is among the most responsive to disruption of nuclear RNA levels. And this was a major clue and really directed our next steps in this project, where we aim to define two fundamental rules rather, for TDP nuclear localization. So rule number one is TDP exits the nucleus by passive diffusion. And this was work that was really spearheaded by Lauren Duan. She's a very talented student who started working with me as an undergraduate and then as a postgraduate. Now she's interviewing for MD PhD programs herself. And she developed this permeabilized cell assay to be able to experimentally isolate and manipulate TDP-43 nuclear export. So in this assay, she was essentially able to pop open cells, leaving the nuclear membrane intact, and then monitor TDP-43 nuclear export in a variety of different conditions. And so using this method, along with a lot of other confirmatory studies, Lauren was able to show that TDP-43 doesn't use any specific carrier protein or energy-dependent pathway to leave the nucleus. In fact, it simply just diffuses across the nuclear pore, which are these aqueous channels embedded in the nuclear membrane through which the nuclear transport occurs. And so this begs the question, if TDP-43 can simply just diffuse out of the nucleus, then what accounts for the fact that it normally is predominantly concentrated in the nucleus? So how does that concentration gradient get established? And that leads me to the second rule, which is that RNA sequesters TDP-43 within the nucleus. And so this work was done in collaboration with a long-term collaborator of mine, Petr Kaleb. He's a cell biologist and a nuclear transport expert who's in the Department of Biomedical Engineering here at Hopkins. And using a variety of clever methodologic tricks, including digesting the RNA within the nucleus using RNase A, and also putting in little decoy segments of synthetic RNA oligomers, as shown over here, we were able to determine that TDP-43 residence within the nucleus critically depends on the availability and binding to its nuclear RNA targets. And so taken together, these studies are really supporting this model where TDP localization within the nucleus depends on binding to these GU-rich or guanine-uridine-rich RNA sequences. These are right near the splice sites where TDP performs its critical splicing modulatory function. And so there seems to be this intrinsic link between TDP's normal function within cells and where it's localized. And binding to those RNAs is what determines whether it's available to freely leave the nucleus or not. And so this finding has major implications then for our overall goal of trying to understand TDP mislocalization in disease, and raises the question of whether a disruption of nuclear RNA metabolism or localization could be one of those upstream factors leading to mislocalization in disease. And then also raises the idea of developing RNA-based strategies to rescue the mislocalization. So those are the type of studies we're actively pursuing now in the laboratory, more complex models. We're also examining postmortem tissue, as shown here, that we're fortunate to have through Target ALS, as well as the generosity of our patients. These tissue-based studies, we're developing some RNA-based probes and also looking at the gradients of TDP-43 and other RNA binding proteins compared to RNA. So just to show you some examples of some of this staining, this is TDP-43 and some of those large upper motor neurons in the motor cortex. And you can see in this patient who died of a non-neurologic illness, TDP is in the nucleus where it's supposed to be, along with this nucleoporin protein that's in the nuclear pore complex and another RNA binding protein that's strongly nuclear as well. So now that we know which RNA binding proteins are RNA sensitive, like TDP-43, we're screening for their co-mislocalization in disease, as well as looking at RNA gradients in these cells that you can see from a C9orf72 case where TDP-43 is out in the cytoplasm. And so looking for any correlation that would support this idea of an RNA gradient-based event. And we're also working on multiple strategies to promote TDP-43 nuclear localization based on our finding that RNA tethers TDP within the nucleus. So the first of these approaches, shown here, utilizes modified RNA bases. These are base analogs that either can be incorporated into the host cell RNA or delivered in short oligomers. And this approach has been quite successful in our basic shuttling models. So we're now working with collaborators to test in flies, and also working with Johns Hopkins Drug Discovery to optimize the chemistry and try to improve specificity and toxicity. The second idea is sort of design specific nuclear retention sequences that allow TDP-43 to bind, but also are connected to a motif that would promote nuclear retention of the entire construct. And lastly, we're also looking at approaches that would modulate endogenous RNA processing to try to boost the availability of the normal binding sites for TDP-43. And these last two approaches are much more in preliminary stages, but we're trying to have multiple shots on goal in terms of being able to augment nuclear TDP-43 RNA binding. And so I'll stop there and thank members of my lab, as well as Jeff Rothstein's lab, who has been my faculty member, and my faculty mentor as I've been trying to set up my own lab, as well as Johns Hopkins Drug Discovery and multiple collaborators at other universities. And I particularly would like to thank all of our funding sources, including the NIH and the Packard Center here at Hopkins. So I'll stop there and I'm happy to take your questions.
Thank you so much, Dr. Hayes. We don't have any questions right now. Just give it a few seconds. All right, well, I don't see any questions right now, but if you have questions, you can type them in and we can do them at the end.
Happy to.
With Dr. McCray's presentation as well. Again, thank you so much, Dr. Hayes. Now I'm going to introduce our last speaker, Dr. Brett McCray. Dr. McCray is a physician scientist with a clinical and research focus on Charcomary tooth disease, the most common form of inherited neuropathy and the most common inherited neurologic disease worldwide. He has a basic science lab that focuses on the pathogenesis of inherited forms of peripheral neuropathy with the goal of identifying common pathogenic mechanisms and novel, broadly applicable therapeutic targets. Hand it over to you, Dr. McCray.
Thank you, Rachel. So let me just get my screen sharing going here. Are you able to see everything?
Yes.
Sorry, just 1 second. Get the laser pointer. Okay. So we're going to switch gears a little bit away from ALS and talk more about peripheral nerve disease and also nerve injury. And maybe how this gene TRPV4 might play a role. So first, I'd like to thank everyone for attending and for the opportunity to talk today about our work on TRPV4. Hopefully people will find it interesting. So first, I want to step back a little bit and talk about what's a pretty large problem in neurology in general. And that's there's really a treatment vacuum when it comes to treating people that have nerve injuries from various causes. So nerves can be injured in a number of different ways, whether it's stroke or trauma to the brain or spinal cord, trauma to peripheral nerves, or exposure to things that are toxic to nerves. And so we do know a lot of what happens downstream of nerve injury. There's a lot of pathways that get activated. There's swelling, there's breakdown of what's called the blood-nerve barrier, inflammatory cells invade the area, and there's release of things that can be toxic. And so we do understand some of these pathways, but we still don't have any treatments to really target these pathways to provide either protection to the nerves or to hasten their recovery after these kinds of injuries. So the focus of my lab is really to study inherited neurological conditions. And so the idea is that inherited neurological diseases are sort of like nature's experiments. And what they really do is focus our attention, we think, on disease genes that are likely to be really important for nerve function. So if a mutation in a particular gene causes nerve disease, that really tells us that it's important for overall nerve health. And so one really large class of inherited nerve diseases is what's called Charcomary tooth disease. And so this is a group of conditions that are related that cause inherited peripheral neuropathy. And so this collection of conditions has the feature of causing progressive loss of strength and loss of muscle, usually in the lower extremities, leading to this sort of thin leg appearance and changes in the way the feet look, and also some loss of sensation. And so often later in the course of the disease, there's also weakness that affects the hands and leads to atrophy of the muscles in the hands. And so to date, there's over 100 different genes that have been identified as causes of CMT. And so you see on the right just a partial list of some of these genes. And so over the past 20 to 30 years, people have been studying the pathways related to these genes in a lot of detail, and they've really highlighted a number of pathways that seem to be important for nerve health. But there's also some outliers in this list of 100 genes, and one of them in particular is this gene TRPV4. And so this is a cause of neuropathy that my mentor, Charlotte Sumner, discovered about 10 years ago. And she immediately recognized that TRPV4 has many sort of unique properties as compared to other CMT genes that really suggest that it might be a good therapeutic target. So I'll go into this in more detail, but briefly, it's expressed on the surface of cells. If you get rid of it completely, it doesn't cause disease. And there are drugs that can readily target this particular protein, TRPV4. So that's been the focus of my work and a lot of her work as well. So what is TRPV4 neuropathy? So this is a group of conditions that are caused by dominant mutations in TRPV4 that lead to various forms of neuropathy. The most predominant symptom is weakness of the arms and legs, and there can also be sensory loss. And so these TRPV4 neuropathies are kind of grouped into 3 different types. One type has sensory and motor dysfunction, and then the other 2 types are purely cause problems with the motor nerves. Somewhat similar to ALS that you heard before, but has a very different progression. And so there's also what we call a congenital onset variety, which means that the onset of the weakness and difficulties with movement occur at birth. Another really important feature and somewhat unique feature of TRPV4 neuropathy is patients that have these mutations develop weakness of their vocal cords and often also weakness of their breathing muscles, in particular the diaphragm. And so the vocal cord weakness is a pretty big problem. And I just wanted to go into a little bit of background about what the vocal cords do and how we assess them. And so these can be visualized using a camera basically inserted through the nose. And then the vocal cords are these white strips here in the picture on the right. And so this is just a video showing kind of what normal vocal cord movement looks like. So when you breathe in, the vocal cords open to allow air to flow in. And when you speak or sing, the vocal cords come together. And so you'll see with this video, this person takes a breath initially, and then sort of sings. You see the vocal cords come together. And then at the end of the video, she takes another breath in and the vocal cords open. So in TRPV4 neuropathy, patients have weakness of the opening of the vocal cords. And so this is a 3-year-old girl who had severe weakness of her vocal cords. And what you can appreciate, if I can get this, sorry, to play. She has this finding that we call stridor, which is a very loud noise when she breathes in. Sorry. So you can hear this, what we call stridor, and you can see she's working really hard to breathe. And then at the end of the video, you'll see that she has a laryngoscopy procedure where you can see the vocal cords are not moving at all. They're completely paralyzed. So she's breathing through this very narrow airway here. She actually needed to, unfortunately, have a tracheostomy because of this airway obstruction. So what is TRPV4 and how does it cause disease? So TRPV4 is an ion channel that sits on the surface of cells. So this is the 3-dimensional structure of the channel. And basically it creates a pore that allows things to flow into the cell when TRPV4 opens. It's expressed in a number of different tissues in the body, somewhat in the nervous system, although less than we'd expect given it causes this neurological disease. It's also expressed in the lungs and kidney, and also the cells that line blood vessels. So the way it works is it is stimulated by various conditions to open, and then it allows things to flow into the cell. In particular, it allows calcium ions to flow into the cell. And the mutations that cause disease, when we've studied them in the lab, all seem to increase the opening of the channel. So they allow too much calcium to flow into the cell. So you see here, the mutant channel on the right allows more calcium in, and that seems to be really critical for what happens in the disease. And one of the reasons we're so interested in studying this disease and studying TRPV4 in general is that we think that blocking TRPV4 could be a really effective treatment for patients. And there's actually already a really good TRPV4 blocker drug that has been in clinical trials for other indications. It didn't work in these other trials, but it was shown to be safe. And we think potentially we could try this drug in patients that have TRPV4 mutations, and it might have a therapeutic benefit. So we wanted to learn more about sort of what goes wrong with TRPV4 mutations and how it causes disease. And we looked at the structure of the protein. And what we found is that the mutations that cause neuropathy tend to cluster in one part of the TRPV4 protein, what's called the anchor and repeat domain, shown here on the right. And so the anchor and repeat domain shown here on the bottom is this red area, and the neuropathy causing mutations are shown in blue. And what we knew already was that the anchor and repeat domain is thought to be important for binding to other proteins and regulating the way TRPV4 opens. So we did a screen to look at things that might bind to this region and might be disrupted by neuropathy mutations. And what we found was this protein called RhoA. And through a lot of work, what we showed is that RhoA seems to be really binding directly to this area where all these neuropathy mutations are occurring. And so we went on to show that the mutations that cause neuropathy completely disrupt binding of RhoA to TRPV4. And that has the effect of overactivating RhoA, which we think may be important in disease. So what is RhoA? It's known as a master regulator of the cell cytoskeleton. So the cytoskeleton of cells, you can think of as sort of the scaffold that gives cells their shape. And what RhoA does is sort of make the cells more stiff and kind of make them contract. And so it's been well studied already, and it has a known role in promoting nerve growth. It actually inhibits nerve growth. So when RhoA is active, the axons and dendrites that Dr. Berth mentioned in the first talk tend to grow less. And when RhoA is inactive, they tend to grow more. It's also shown to be really important in regulating the blood-brain barrier, which is basically a barrier formed by blood vessels that keeps things circulating in the blood from leaking out into the brain and spinal cord and nerves where it can cause problems. And so again, inactive RhoA seems to promote blood-brain barrier function. And then when RhoA becomes activated, it sort of pulls apart these barriers and allows things to leak out into the nervous system that aren't supposed to leak. So to summarize a lot of data, what we went on to show is that mutations in TRPV4 block binding to RhoA, which leads to increased activation of TRPV4, so more calcium coming in, and then also activates RhoA. And so RhoA gets overactivated, and this changes cells in a way that tends to cause them to sort of contract. And for neurons, it makes them sort of shrink. So we then wanted to test some of these insights in a model system. And so we turned to a fly model of TRPV4 neuropathy that was made by Dr. Tom Lloyd's lab, who is the mentor for Dr. Berth. And so when we put mutant TRPV4, the disease-causing forms of TRPV4 in flies, we saw a number of changes. So the fly wings fail to open, the flies don't crawl very well, and we also see degeneration of the nervous system. So this is a fly larvae, and on the top is sort of the equivalent of the brainstem of the fly, and on the bottom is sensory neurons and dendrites. And so we see when we put mutant TRPV4 into these flies, the axons degenerate and also the dendrites of the neurons degenerate. And importantly, if we give a TRPV4 blocker drug known as GSK219, so this is different than the one I showed that was in the clinical trial, but it's sort of a similar structure. This can rescue this degeneration of these neurons, suggesting really that the opening of the TRPV4 channel is really important to causing disease. We also then looked at the importance of RhoA in this model. And what we found again was that if we block RhoA, we can rescue the axonal degeneration as well as the degeneration of dendrites. So this work really highlights both the opening of the channel as an important part of the disease and also RhoA activation downstream of that. We then wanted to test these things in a mammalian system. So Jeremy Sullivan and Charlotte Sumner's lab generated this knock-in mouse model of TRPV4 neuropathy. And so basically what was done was to engineer a human neuropathy causing mutation into the TRPV4 gene in the mouse. And these mice developed this really remarkable rapid onset weakness that progresses to paralysis and death within a couple of days. And so when these mice have 2 copies of this disease gene, they die within 3 weeks of life. But again, if we treat them with this TRPV4 blocker drug, GSK219, we're able to completely rescue the lethality. So I want to show you a video of what these mice look like when they're getting sick. Hopefully I can get this to work. A little trouble with my pointer. Sorry, everyone, bear with me for a second. There we go. So this is a sick mouse, one of these knock-in mice. And so it's having a hard time lifting its front paw. And you can see it also has difficulty lifting its head. So it's having a lot of weakness and difficulty walking. So it's able to walk a little bit, but it's very weak. And if this mice weren't treated in some way, it would die within 1 or 2 days after this point of disease. And so in this slide, this is not the same mouse, but it's a mouse that was at a very similar stage of this weakness phenotype. And it was treated with this GSK219 drug. And then 12 hours later, this mouse is much better and running around and able to sort of get out of the cage, and then has to be grabbed by Jeremy, who's taking this video. So it's really remarkable and rapid recovery of this phenotype with this GSK219 blocking drug, which is really exciting to us. So the other question we wanted to ask is, how much do these mice really look like what human patients have? And so I mentioned the importance of this vocal cord weakness to these patients. And so we looked at the vocal cords of these mice. So first, this is just a normal mouse with the vocal cords opening and closing as it breathes. And on the right, we look at one of these mice that's sick, and we see the vocal cords are moving very poorly. So it does seem that these mice have a lot in common with what patients experience. And so altogether, this work from the flies and from the mice really suggests that TRPV4 neuropathy might be something we can treat with the available drugs that are out there already. So to try to make that happen, we've been focusing a lot of work on trying to move things towards a clinical trial. And there are some barriers to that we're trying to address. So one of them is that there aren't that many of these TRPV4 neuropathy patients out there. And we don't really know enough about sort of what happens naturally in this disease without any treatment. And so we're developing a TRPV4 patient registry through the Inherited Neuropathy Consortium. And the goal is to help to learn more about these patients and identify patients that might be good candidates for treatment someday. We've also started a TRPV4 channelopathies clinic at Johns Hopkins. And so this is a subspecialty clinic within our existing Charcomary tooth disease clinic. And we brought together a multidisciplinary group of clinicians to really try to boost understanding of what happens with these patients and look for things that we might be able to use to monitor whether drugs are effective in a clinical trial. So all this work I've been talking about really suggests that for patients that have mutations in TRPV4, we think that TRPV4 blocking drugs may really be an effective therapy, and we're working towards making that happen. But the other implication of this work is that blocking TRPV4 might have other therapeutic applications. And so there's emerging literature that TRPV4 seems to be overactivated following strokes, following traumatic brain injury and spinal cord injury. So we think TRPV4 blockers in that setting might also be protective. And then there's also literature about the role of RhoA in a number of different neurological conditions. So RhoA gets overactive and blocks nerve regeneration following injury. And RhoA is also overactive when there's blood-brain barrier breakdown, which also happens in things like stroke, traumatic injury, et cetera. And so what we think with this work is that we may be sort of connecting the dots among these different findings, and that it may be that TRPV4 is sort of a signal to overactivate RhoA, and that contributes to a lot of downstream damage that happens after nerve injury. And we think potentially that blocking TRPV4 might really be able to help in a number of different nerve injury conditions. And so to explore that further, we're doing a number of things in the lab. So we're looking at TRPV4's role in peripheral nerve injury. We're doing this in mice by using a model where we crush the sciatic nerve, which is a nerve in the hind leg of the mouse, to see whether manipulating TRPV4 can be protective. We've also, with a collaborator, Barrington Burnett, looked at a little bit at traumatic brain injury. And what he's found is that if we give this GSK219 TRPV4 blocker to mice that have undergone traumatic brain injury, it does seem to protect from sort of secondary injury following that initial injury. And we're also planning to look at the role of TRPV4 inhibition in chemotherapy-induced neuropathy. And so this is a really important and unfortunate side effect that can happen with chemotherapy to treat cancer. We know that a lot of people develop neuropathy, and we think that inhibiting TRPV4 might be something that could potentially limit the development of neuropathy with chemotherapy exposure. So with that, I'd like to acknowledge the many people who have contributed to the work that I presented. So Charlotte Sumner has been my mentor and is helping me in this process of establishing my own lab. Jeremy Sullivan did the bulk of the work characterizing these amazing TRPV4 knock-in mice. As I mentioned, the Lloyd lab, in particular Brian Woollums, did a lot of work with generating and characterizing this TRPV4 neuropathy fly model. And then I also acknowledge Barrington Burnett, who helped with these traumatic brain injury studies. And so I'll finish up there and we can take questions.
Thank you so much, Dr. McCray. We do have two questions for Dr. Hayes back from her presentation. So I'm just going to do those really quick. Could cytoplasmic localization of TDP43 be due to structural issues?
I'm reading the question again. I'm trying to read the question again, but I can't find it. Structural issues. So there are folks at Hopkins and other places who've identified some structural changes in the nuclear pore complex itself, which could conceivably alter the distribution of cargos that traffic in and out of the nucleus like TDP43 does. Currently, the data do not suggest that that's probably the explanation for why it's mislocalized, for two reasons. One being that in those studies, specifically some of the studies that the Rossing lab has done, they've tested whether or not the abnormalities in the structure of the nuclear pore that they found made them leakier. And the answer seemed to be no, that they weren't leakier. So that's one reason I suspect is not the case. The other reason is that if there was a structural problem, you would expect there to be this global misassortment of all sorts of cargoes that traffic in and out of the nucleus being misplaced. And that's really not the case. In fact, I showed you some of the staining we've done to look at various other cargoes that shuttle like TDP43 does. And thus far, we have not found anything that co-mislocalizes with it. Even those proteins that use the exact same receptors to get into the nucleus seem fine. So obviously, you can never say never because we still haven't pinpointed the exact cause of it, but the jury's still out, but it's not clear that there's a link so far.
Okay, great. Thank you so much. And one more question for you, Dr. Hayes. Why is it so hard to diagnose ALS?
That's a good question. I mean, so two answers. One is that once people have developed the full-blown constellation of symptoms of ALS, it's actually a very easy diagnosis to make, meaning that once they have involvement of upper and lower motor neurons, and the course of the disease has clearly shown progression, and a trained neurologist such as the other colleagues on the call can see the person in clinic and have a very high suspicion without really much follow-up testing that unfortunately this is ALS. When it's right at the beginning, it can be very difficult to know because it can present, as Sarah mentioned, with simply cramping in the muscles or twitching of the muscles or some vague kind of weakness or incoordination of just one limb, a hand or a foot. And so in that case, especially when it's early, just a few months in, you have to kind of think broadly, make sure that you're being comprehensive about looking for potential causes of this. And sometimes we do struggle to see changes on an EMG or electrophysiology at that point. And so at that point, it is tough. We tend to kind of have to wait and watch and see how symptoms progress over time. But what we really need, and there's a lot of work going into this, but we're just not to that point yet, is a biomarker, a blood test, some kind of diagnostic test that we can use to tell patients yes or no, to expedite getting them into clinical trials, getting them on treatment early. And right now, we just don't have one that's ready for diagnostic use. And so the C9orf72 gene test is really about the only thing that we can use. As I mentioned, it's about 1 in 12 cases. And so most of us in our clinic here at Hopkins will test everyone for that, because if you see that combined with even mild symptoms, you have a diagnosis. But in those patients who test negative, sometimes that early phase, there's a lot of questions. And I know it's very tough on patients as well, that sort of odyssey of waiting for diagnosis.
Thank you so much, Dr. Hayes. Dr. McCray, we also have some questions for you. Do you expect that TRPV4 antagonists would be beneficial for other CMT patients, not only for TRPV4 mutation carriers?
Yeah, maybe. I think the core question is, when TRPV4 is sort of active, it seems to do a lot of bad things for the nervous system. And the question is TRPV4 likely to be active in other forms of CMT? And the things that tend to activate TRPV4, so it's sort of mechanically sensitive. So it's activated by stretch and pressure and things like that, which is why we're kind of focusing on nerve injury. We don't have a great sense of where it's expressed in the nervous system, actually. It doesn't seem to be that much present in nerves, in neurons. But it's certainly possible that there's some cascade of events that happens in other forms of CMT that would activate TRPV4. And in that case, it could be helpful to inhibit it. But again, I think the question is, is it active in certain disease states or not? And if it is active, then inhibiting it would be helpful.
Is there a treatment for foot neuropathy?
So really the first question in any form of neuropathy is the cause, and the treatment options depend on the cause. And so it can be, cause can be genetic, as in Charcomary tooth disease, or related to a number of other things like diabetes or vitamin deficiencies. So it really depends on trying to find the etiology or the cause of it. And unfortunately, a lot of times we do sort of routine workup and even more extensive workup to understand the cause of someone's neuropathy, and we don't find anything. That happens at roughly a third of the time. We call it then idiopathic, which just means we don't know why you have neuropathy. And in that case, there's no generally helpful treatment that everyone can get to protect nerves. But that's the kind of thing that we were looking for when we do these, this research pursuit of trying to understand genetic forms of neuropathy with the hope that we identify targets that might benefit people that don't have genetic forms of neuropathy. But we're not there yet. We don't have anything yet that we can give everyone to help with all forms of neuropathy. It's a big treatment hole.
We have a similar question, which I assume is going to have a similar answer, but I'm going to ask it anyway. A patient with very specific symptoms, peripheral neuropathy for over 5 years with burning sensations, numbness, general heaviness in lower legs and arms, no weakness in hands or legs, tested for ALS, MS, and other autoimmune diseases in the past month or so, has had general pins and needles feeling in the face, arms, and chest. Basically, I know that you just said it's not always a hereditary condition, but, do you have any recommendations or treatment?
Yeah, I think one thing that's puzzling to a lot of us is that, so there's inherited neuropathy where there's a specific gene that causes it, and those are being studied a lot, and we're learning a lot about those. But there's a lot of people that have neuropathy where we don't have a clear cause. We suspect that there is some genetic component to those, that it may not be one particular gene, but it might be several sort of risk factors. And so there's a lot of work to try to understand those risk factors. And we're even finding new genes that cause inherited neuropathy that kind of hint at risk factors. So for example, there's a gene that was recently found that leads to accumulation of this sugar called sorbitol, and that causes inherited neuropathy. But it's also been known that sorbitol is something that accumulates in patients with diabetes. So I think we're starting to see this sort of overlap between inherited genetic neuropathy and diabetic or idiopathic neuropathy, and we're starting to kind of learn that there are not just single genes that cause neuropathy, but probably other genes that convey susceptibility. And so there's a lot of research interest in trying to sort this out and hopefully be able to tell people more definitively why they have neuropathy, what we can do about it.
Thank you. Is the GSK drug brain penetrant, and do the mice have non-neuronal phenotypes?
Yeah, it's a great question. So it is not brain penetrant. That's something that the drug companies are interested in working on. So, which will then lead to the question, how are these mice getting rescued if this is a neuronal disease and the drug is probably not getting to the brain or the neurons? And so we actually think this might be due to TRPV4 in the blood vessels and a specific function there. And so that we know that the drug can get there. Was there the second part of the question? I forget now. Hopefully that answered it. It's a great question, though.
Do the mice have non-neuronal phenotypes?
Yeah, so one thing I didn't mention is that there are mutations in TRPV4 that also cause bone diseases. And in some cases, most of the time they're non-overlapping. So you either have nerve disease or you have bone disease, but there is some overlap in some patients. So interestingly, the mice do seem to have a little bit of a bone phenotype where the snout looks a little bit abnormal. But otherwise, we've looked pretty extensively, or mostly Jeremy Sullivan has looked pretty extensively, doesn't see other clear phenotypes in these mice. So it seems to be a really nervous system specific effect, which is interesting.
Okay, thank you so much, Dr. McCray. I have one more ALS question. If Dr. Berth or Dr. Hayes could hop back on. Is there evidence that trophic factors from the muscle of Schwann cells are involved in ALS, or is the disease thought to be only intrinsic to neurons?
I can take this. So it's not just the motor neurons that are affected. So we know that that microglia and astrocytes are affected as well. And it's kind of hard to say like if this is like upstream of the motor neuron involvement, or if it's kind of in a more like a reaction to motor neuron degeneration. Schwann cells, I think historically it's thought that the Schwann cells control myelination of nerves. So when there's this degeneration of the motor nerves, then the myelination can become impaired. But there have been recent studies looking like the Schwann cells that produce the myelin can actually perhaps release trophic factors to kind of coordinate the inflammatory response. So I do think there may be a role for Schwann cells, but there is a lot more research to kind of parse that up.
Thank you so much, Dr. Berth. And I just want to, before I hand it back to Jamie to close this out, I just want to thank Dr. Berth, Dr. Hayes, and Dr. McCray so much for sharing your research with us. It's truly fascinating. And with that, I'll hand it over back to Jamie.
This was wonderful. We totally appreciate everybody's time and energy, and we hope you really enjoyed our panel. Thank you to the speakers again for your time and talent. And we'd also like to thank all of you, our audience, for joining us for today's webinar. A follow-up email will be shared with you following the webinar that will include a recording of the program along with a list of resources and our event schedule. And we hope that you will join us at an event soon. So with that, I hope you all stay healthy, happy, and safe. Thank you so much.